Hello, everyone, and welcome to the lecture on azo-dyes. Okay, so what are azo-dyes? Azo-dyes are organic compounds that contain the functional group R bonded to nitrogen, double bonded to nitrogen, bonded to another R. So you can see, for example, that functional group displayed in this azo-dye. And very commonly, both R and R prime are usually some sort of aromatic ring with a variety of substituents attached to them. Azo-dyes are synthetic in nature, meaning they're synthetically prepared as opposed to naturally found dyes. And they're widely used to dye cloth and other textiles, leather, and they're often also used as a coloring agent in preparing some foods. So they are highly conjugated. If you notice, there's p orbitals all the way around both rings. Both nitrogens have a p orbital linking the two conjugated rings, and also the oxygen has a p orbital joining the conjugated system. And they often have brilliant colors, like you see here with these compounds in test tubes. To recall that we have talked about how conjugation affects color, and if you want to review that, go back to the chemiluminescence lecture. And then this compound is just shown with one substituent, but there can be multiple substituents on both of the aromatic rings. And the effect of adding additional compounds or atoms with p orbitals is we change the chromophore, which is the part of the compound within an extended conjugated pi system. That will change the wavelength of light that is absorbed, and that will change the color that's reflected in the visible spectrum. Okay, let's talk about some examples of different azo-dyes. The first one here is the structure of methyl orange. Notice the azo linkage and the two aromatic rings. This ring has a dimethylamino substituent, and the nitrogen does have a p orbital, right, with a lone pair inside of it that is extending the conjugation. Another common functional group found in many azo-dyes is the deprotonated sulfonic acid group. The reason why this is so common is because it's advantageous to have the dye be water soluble. So one of the functional groups that can make it water soluble is having a sulfonic acid that's easily deprotonated at most pHs, since it's so acidic. Methyl orange is used as a pH indicator in titrations, just like you may have used phenolphthalein when you were in general chemistry. It's used because it has a clear and distinct color difference when it is protonated versus not protonated. Right? So for example, if we get down to an acidic enough pH, this will become protonated and the color of the compound actually changes. And that change occurs in the acidic region at about 3 to 4 pH. So it's often used in the titration of acids. I added this nifty little chart here so you can see how the color, goes from red to yellow in that pH 3 to 4 range. Phenolphthalein you may be more familiar with. That's the pink to colorless, right? And that occurs in like the pH 8 to 10-ish range. So on the basic side. And then pH paper as well will change colors depending on if it's introduced to an acidic or basic solution. But the litmus paper is actually a mixture of a number of different dyes. I think there's like somewhere between 10 and 15 dyes that are used to make up litmus paper. Another example of an azo-dye is trypan blue. You can see here, this definitely has a very extended conjugated system. It has 2 azo linkages and 4 different deprotonated sulfonic acids. It also has amino groups and phenol groups. So it's a very interesting compound. It is used not only as a dye for dyeing textiles like cotton, but it's also used in the biosciences to stain cells. And specifically, dead cells will be stained blue, and live cells don't get the same amount of stain inside of the cell because of the cell membrane. So for example, it's common if you're going to study biochemistry or biology to need to do some sort of cell culture where you're continuously growing cells. And one of the things that's important to do when you're growing cells is to count the number of cells. So for example, using a magnifying, a microscope, you can prepare a slide of the cells and then count up the number of cells you can see in a number of squares. So notice that the dye gets into cells that have a cell membrane that is permeable because it's lysed or it's dead. Okay, so they're able to count the live cells here, and then these blue ones are the dead cells. I mentioned before that azo dyes are used also in the food industry as a coloring agent. So for example, you may have heard of red number 40. It's also known as Allura Red AC. This is the structure of it here. Again, it has that common deprotonated sulfonic acid group, the azo linkage, and it also contains a phenol and a methoxy group. And then another example is yellow number 6, also known as sunset yellow. You can see the structure here. Again, it contains a different aromatic backbone, right, with the azo linkage, but all very similar substituents that you've seen before. So both of these dyes are used worldwide, and red number 40 is by far the most commonly used red dye in the United States. About 2.3 million kilograms of red number 4 is produced annually, and both of these were approved by the FDA for use not only in food but also in cosmetics and other pharmaceutical drugs. For example, they sometimes use them in the coating, right, which is purely for aesthetic reasons. And these dyes are also sometimes used in tattooings and products such as cotton candy, soft drinks, cherry or lemon flavored products, children's medications, again for aesthetic purposes, and even sometimes in dairy products. For example, like in yogurts. Let's talk a little bit about the history of azo dyes. So it was first discovered, or azo dyes were first discovered in 1863, specifically when chemists named Martius and Lightfoot. They were the first ones to couple diazonium ions, and we're going to talk about what a diazonium salt looks like, and amines, right? And they're talking about chromophores. Remember, chromophores just refers to the portion of the molecule that is covered by the overlapping pi system. So in the same year, another chemist named Peter Grice prepared Bismarck Brown, which is actually the first azo dye to be prepared and used. So I included the structure down here. So they coupled a diazonium salt and an aniline derivative. And by the 1880s, there were numerous other azo dyes that were being developed, including para red and vermilion red. Some of the simplest azo dyes, meaning they have the least number of substituents, for example, like the one I showed on the first slide, those are actually yellow in color. But by changing the structure, right, of the chromophore, you can produce other colors, commonly orange and yellow, but even blues and black dyes can be produced. And they are, because they're synthetic dyes, they're relatively easily prepared. They're actually, I think about 60 to 70% of the dye industry is made up of azo dyes. They're commonly used to, again, color cloth and other textiles. They're used in paints, printing inks, varnishes, plastics, even crayons, and many other products. Let's look at the chemistry behind preparing the azo dye. So the reaction involves what's called a aryl diazonium salt. Right? It's called a salt because it's an ionic compound. And again, an aromatic ring will be attached to what looks almost like nitrogen gas, right? But it's actually bonded to the carbon. And this nitrogen is actually highly electrophilic because it's pi bonded to a nitrogen that has a positive charge. So it can actually act as an electrophile with an activated aromatic compound in an electrophilic aromatic substitution reaction. What I mean by activated is the aromatic compound that's used as the nucleophile needs to be electron rich. So it commonly has groups that are either moderate or strong activators attached to the ring. And again, that will enhance the rate of the electrophilic aromatic substitution reaction. So the nucleophilic attack of the pi bond from the activated ring attacks the electrophilic nitrogen, and electrons move up to this nitrogen through resonance. That gives us the resonance stabilized sigma complex, also known as rhenium ion. It does lack aromaticity, so the ring is currently not aromatic, but it is highly resonance stabilized, so it's not too high in energy and can form fairly readily. And then in the last step, we just have proton transfer by some sort of base. They're showing chloride in this example, but these are commonly prepared in aqueous solution. So the base could also be just simple water molecule. So how did we get the diazonium salt? Right? The diazonium salt comes from a process called diazotization, and it starts from an aniline. The aniline is treated with sodium nitrite and aqueous HCl, and the diazonium salt is prepared. Typically, it's used immediately, so it's prepared what we call in situ and used immediately by either reactions such as the Sandmeyer reactions that we talked about in lecture, or in the azo coupling process. Note that the reaction to form the diazonium salt must be kept at low temperature. And that's because the diazonium salts are very reactive. So to mitigate their reactivity, we keep them at low temperature. This prevents decomposition or reaction of the diazonium salt to prepare something undesired. The active electrophile that reacts with the NH2 group in the aniline is what's called a nitrosonium ion. Nitrosonium ions are prepared in situ and then are reacted with amines such as aniline to form the diazonium salt. So the nitrosonium ion is formed from the reaction of sodium nitrite and HCl. So these two react together to form the nitrosonium ion before the aniline gets involved in the reactivity. So let's look at the process first to form the nitrosonium ion, and then we'll see the reaction of the nitrosonium ion with aniline to produce the diazonium salt. So again, we're starting with sodium nitrite because we need to use nitrous acid in order to form the nitrosonium ion, but nitrous acid is unstable, and so it's better to prepare it in situ and then react it further. So sodium nitrite is a good precursor to nitrous acid. If you mix sodium nitrite with and they're showing HCl, but of course, it's an aqueous solution, it's really hydronium ion. But an acid will protonate it to form nitrous acid. It actually can get protonated a second time to make a good leaving group, which can leave, water can leave, and to form the nitrosonium ion. Nitrosonium ion has two resonance contributors. So you can either see the resonance contributor with a triple bond between the nitrogen and oxygen and a positive charge on the oxygen, or a double bond with the pi electrons given to the oxygen as a lone pair, and this would leave the positive charge on the nitrogen atom. So really the positive charge is shared between the nitrogen and oxygen atom. Even though this is the case, the nucleophiles will always attack the nitrogen, and that's because the oxygen is more electronegative, and it would rather have the full set of electrons instead of a bonded pair of electrons. So again, nucleophiles will come in and attack the nitrogen, and the lone pair will be on the oxygen then exclusively. Okay, so once the nitrosonium ion is formed, then because it's highly electrophilic at the nitrogen atom, nucleophiles such as the lone pair on the nitrogen of aniline can do a nucleophilic attack. At this point, we have what's called a protonated N-nitrosamine. It can be deprotonated by a base in solution such as water to form the neutral N-nitrosamine. If this was a secondary amine and this was an R group, this would be the end of the road. This would be what is isolated, the neutral N-nitrosamine. But we can actually react this further and get to an even more stable product because of the fact that there's a hydrogen here, which can tautomerize and move to the oxygen atom. So the next two steps are actually just a tautomerization. So if you imagine that the two nitrogens are carbon, then this is the carbonyl carbon, also known as the ipso carbon, for example, of an aldehyde or ketone. There was a bond here to a hydrogen or carbon. And then this would be the alpha carbon with a hydrogen attached to it. We know that alpha carbons with hydrogens attached to them, those protons are decently acidic. They have a pKa of around 16 to 20. So they can be deprotonated by a base. And this mechanism for the tautomerization that we're going to draw is going to be acid catalyzed because, again, we are under acidic conditions, so we should draw a mechanism consistent with those acidic conditions. So a tautomerization under acidic conditions would start by a proton transfer. So the carbonyl oxygen would become protonated with the acid in solution. And then we can show if we want resonance from a lone pair on this nitrogen, or alternately, we can just show the base coming in, taking the quote alpha proton and electrons flowing up to the oxygen atom. This leaves us with a nitrogen-nitrogen double bond and an hydroxy group attached to the terminal nitrogen. Because we're in acidic conditions, it's not unreasonable to think that sometimes this oxygen will be protonated. And when it is protonated, you notice that it is a good leaving group, and it can leave in this case because of the lone pair on this nitrogen. So this lone pair can help essentially eliminate or kick out the water as a leaving group. And that leaves us with then the diazonium ion, and the counter ion should just be whatever is used as the counter ion in the acid. Okay, so in this particular experiment, you're going to be watching a video of the production of an azo dye. The two organic components that they use in the video are 2-naphthol and benzocaine. And benzocaine contains the aniline group that will react with sodium nitrite and aqueous HCl to form a diazonium salt. Again, this is usually kept at low temperature to prevent unwanted side reactions. And then an electrophilic aromatic substitution will occur with 2-naphthol. Some things I want you thinking about as you're watching the video, and it also asks you these questions that you can answer in the activity, are to think about why or how the aqueous acid will help solubilize the benzocaine in water. You want to think about the functional groups here. Which of these functional groups makes it so that if we mix this with acid, it will become soluble in water. And then notice in the video, they're going to be dissolving 2-naphthol in water using aqueous sodium hydroxide. So ask yourself, why is the base necessary to dissolve 2-naphthol in water? And then an optional extension question, which we can talk about in office hours, is can you explain the regioselectivity of the electrophilic aromatic substitution reaction? And what I mean by that is if you notice which position on the naphthol ring reacts as the nucleophile, right, to attack the electrophilic diazonium ion, it's actually this position, right, which is called carbon number 1, actually. This is the 1,2-naphthol. The 1 position is the one that ends up reacting. And so, of course, you might know that the OH group is a moderate activator, and so it's an ortho-para director. The para position is blocked, but both ortho positions are not blocked. So then the question becomes, why does it react with this ortho position and not this one? Right? So you may want to think about, right, if it was to react in both positions, what would the resonance stabilization, right, or how many resonance contributors and what do they look like, and how would they differ from attack at both of those ortho positions? And think about what's the most stable sigma complex or rhenium ion, and then that should lead to the major product. Okay, that's all I have for the pre-lab lecture. I will see you guys after you watch the video and start working on the activity. Hope to see you in office hours soon.